Muy buenos días a todos, colegas. Un saludo desde el auditorio del Centro de Ciencias Genómicas de la UNAM. Es para mí un gusto, y creo que varios de los colegas también, volver a esta nuestra sede de los seminarios, y aunque sea con pocos asistentes, pues es un avance de lo que hemos tenido los últimos meses y años. Saludos a todos los que se unen a nosotros por métodos virtuales. Es un gusto presentar al ponente de hoy, el doctor Jesús Montiel González, quien es uno de los candidatos preseleccionados para ocupar una plaza de investigador en el programa de investigación genómica funcional de eucariotes en este centro. Jesús hizo su licenciatura en la Benemérita Universidad Autónoma de Puebla, su licenciatura en biología. Hizo posgrado, lo hizo en el programa de ciencias bioquímicas en el Instituto de Biotecnología con la asesoría de la doctora Carmen Quinto, habiéndose graduado de doctor en 2012, cuando fue distinguido con la medalla Alfonso Caso. Desde entonces, el área de interés de Jesús ha sido la asociación de plantas con microorganismos, en particular la simbiosis fijadora de nitrógeno de plantas leguminosas con rizobia, habiendo cubierto durante su carrera diferentes ángulos de este proceso tan relevante para la biosfera. Durante su posgrado, Jesús analizó la señalización de eventos tempranos de la simbiosis frijol-rizobia, en particular el papel de las NADPH oxidasas y de la peroxidasa RIP1 de frijol. Después de su graduación, Jesús realizó una estancia postdoctoral corta, como de 9 meses, en el grupo del doctor Joseph Dubrovsky del Instituto de Biotecnología, y de 2012 a 2017, Jesús fue investigador postdoctoral en el Biological Research Center of the Hungarian Academy of Sciences en Szeged, Hungría, con la asesoría de la doctora Eva Kondorossy, una de las pioneras de la fijación biológica de nitrógeno con gran reconocimiento internacional. Allí Jesús analizó la diferenciación terminal de bacteroides en los nódulos de leguminosa del clado conocido como Inverted Repeat Lacking Clade, publicando sus contribuciones en revistas de prestigio como el Proceedings of the National Academy of Sciences. De 2017 al 2018, Jesús se incorporó como investigador postdoctoral en el Centro de Biotecnología y Genómica de Plantas de la Universidad Politécnica de Madrid, en España, con la asesoría del doctor Manuel González Guerrero, en donde analizó la homeostasis de metales en nódulos de la leguminosa medicado truncata. Y desde 2018, Jesús ha sido investigador postdoctoral en la Universidad de Aarhus, en Dinamarca, con la asesoría del doctor James Stubart, uno de los líderes actuales del área de fijación de nitrógeno, con un grupo con grandes recursos e investigación de frontera, y que participa en proyectos internacionales de gran envergadura para el área, como por ejemplo el proyecto conocido como ENSA, que es European Symbiotic Nitrogen for Africa, financiado por la Fundación Bill y Melinda Gates, entre otros. Aquí Jesús ha explorado los componentes moleculares que determinan la colonización intercellular del rizobio en las leguminosas. Es un proceso ancestral, pero muy poco conocido. Las contribuciones científicas de Jesús han sido publicadas en 18 artículos en revistas internacionales de prestigio que han recibido 455 citas. Además, durante su carrera, Jesús ha interesado en asuntos de docencia y formación de recursos humanos, ha asesorado a estudiantes de diferentes niveles y ha participado en la elaboración de donativos para proyectos de investigación. Es pues un gusto tener aquí a Jesús, quien algunos lo conocemos desde su posgrado, y ahora como científico joven de gran calidad le damos la bienvenida y escucharemos su seminario. Y aquí está el título del seminario de Jesús, que es Lotus IRBG 74 Symbiosis, a novel working model to explore intercellular colonization and to expand our knowledge to symbiotic associations in common bee and peanut. Bienvenido, Jesús. Gracias.
Well, first, I really want to thank you for giving me the opportunity to share with you the research I have made in the last years. And also, I am very happy to be here in this auditorium because the last time I was here, it was to defend my PhD thesis. And now I am back here again. So, well, first, I want to So I want to tell you that my talk is divided in two sections. So first, I want to tell you about the lotus IRBD symbiosis. And here I will show you the characterization I made about this process and the different approaches I have been using in the last 3.5 years to identify postdocs and many PhD students, lab managers, technicians, and learners. And all the research projects we are conducting in this laboratory are focused in these 3 points. Engineering nitrogen fixation. Also, we are trying to understand how these symbiotic receptors are working. And also, we are trying to develop modern breeding methods. And well, just to emphasize how important is this symbiotic field in the Nordic countries and in Europe in general, I want to show you the different grants that our group has obtained in the last 9 years. And these different grants are from different scientific agencies. So let me show you. So from these different scientific agencies, and especially my research project is supported by the Bill and Melinda Gates Foundation. And this project is part of a very big project called ENSA that Ina already told you about. And the ENSA project participate many of the researchers, of the most recognized researchers in the field, in the leading rhizomia symbiosis field. And they belong to different research centers and different universities, some of the most prestigious universities in Europe. And this is a very big collaboratory network with all these scientists involved in the ENSA project. And we are analyzing different aspects of the legume rhizobia symbiosis. And well, in my case, I am working with Lotus japonicus. And Lotus japonicus is this legume here, and is able to establish symbiotic associations with rhizobia for the development of the nitrogen fixing oil. Also, Lotus established symbiotic association with Rhizophagus irregularis for the arbuscular mycorrhizal symbiosis. And recently, we are also using Lotus japonicus to study microbiome associations. And there are several reasons why we are using Lotus japonicus. And these are some of the reasons. And here I am just highlighting some of the fronts that are very linked to my research projects. But in general, all of them are very important. And well, as you know, the legume rhizobia symbiosis starts in the rhizosphere with a chemical crosstalk, where the legumes release flavonoid compounds, and some specific flavonoid compounds are perceived by the rhizobia. And once they are perceived specifically, they start to synthesize and to release the non-symbiotic nodulation factors. And these node factors are lipoketo-oligosaccharides that are specifically recognized by the legumes, and especially by the root hairs. And if the compatibility is good and the recognition is efficient and successful, then the rhizobia will attach to the root hair tip, and then there is a root hair correlate. And at this point, there is a very important process, because the rhizobia enters into the root hair cells via a tubular structure called infection thread. And this infection thread migrates into the epidermal and cortical cells. In parallel, there is cortical cell division, giving rise to the nodule primordium, and this will give rise to a nitrogen-fixing nodule. And in the nodule cells, the bacteria are released from the infection threads, and they can start to fix the nitrogen. And well, here we have in legumes two types of nodules, the indeterminate nodules and the determinate nodules. And I will tell you more about it in the talk. Well, most of these knowledge we have obtained using the model legumes, like Medicago truncatula and Lotus japonicus. And these model legumes have this intracellular colonization via root hair infection threads, the one I told you already. However, rhizobial infection is a bit more complex, and there are several examples in nature about a different mechanism of rhizobial colonization. And we call this one intercellular infection. And there are different modalities of intercellular infection. So in one case, the bacteria can colonize between the cells, as you can see here in this drawing. There is another type of intercellular colonization, and it's via crack entry during the emergence of lateral roots. And actually, this intercellular colonization is widely spread in the nodulating legumes. And it is estimated that around 25% of all legume genera are intercellularly colonized. And here I show you some examples of the different species that are intercellularly colonized, and they are widely spread. And there is some evidence that this is probably the ancient mechanism that rhizobia employ to infect legume roots. And also, there is some kind of evidence that this is a more simple method to reach rhizobial colonization in the legumes. But unfortunately, we know very little about this process, because this process is observed in legumes that we don't have enough resources to study the symbiotic process. So it is very important to develop a working model to study the intercellular infection, because this has the potential to engineer this process and to understand better this process. And well, so we know that in Lotus japonicus, the model legume Lotus japonicus, in some mutant backgrounds, like the node factor receptors mutants and other receptor mutants, they can form sometimes nodules, but in a very low frequency. And when these nodules are formed, This is just a nodule slide. You can see that the bacteria colonize the epidermal cells intercellularly. Here also there is another example of a different mutant background where the rhizobia starts to colonize the nodule cells intercellularly. But this is in a very low frequency. However, there is a legume, Sesbania species. They can be intercellularly colonized. And here you can see an example of the colonization by a very interesting strain, IRBD74. And in this case, you can see the deposition of IRBD74 during the emergence of these lateral roots, and eventually they will form the nodules. And IRBD74 is very interesting because it's actually an agrobacterium strain that belongs to the Rhizobiaceae. And they have found that even IRBD can colonize Arabidopsis as an endophyte. And even there is some evidence that IRBD74 can colonize rice roots. So it's a very efficient colonizer. So with all this background, we decided to inoculate Lotus japonicus with IRBD74. And luckily we found nodules. Here are some representative images at different weeks after inoculation. And you can see that the nodules are pink, so it's an indication of nitrogen fixation. And also the aerial part is green, so the plants can grow. So we decided to analyze in more detail this process. And first we decided to observe the infection process by confocal microscopy. First, here I show you a typical example of a root hair infection in Lotus japonicus. Here you can see the emergence of a nodule primordia with the cortical cells here, and the colonization of Mesorhizobium via this root hair infection that migrates to the epidermal cells and also the cortical cells. And this can be observed at 1 week post-inoculation. However, when we observe lotus root with IRBD at 1 week post-inoculation, we didn't see any root hair infection threat. And actually, we saw the IRBD only on the surface of the roots over here. And when we analyzed the roots at 2 weeks post-inoculation, we could see an emerging nodule primordia. Maybe it's not so evident here, but you can see the emerging nodule primordia and not root hair infection threats. But we can start to see some evidence of intercellular colonization around here. So we decided to analyze, like in more detail, this process. And here is just a magnification of a nodule primordia of Lotus japonicus with IRBD74. So here in this part, here are the epidermal cells. This part is the epidermal cells. And I hope you can see in this presentation, but here there is the IRBD effect in the epidermis, and then it starts to colonize intercellularly and forming these infection pockets. And from these infection pockets, you can see that IRBD starts to infect the lotus roots intercellularly over here. And we decided to follow this infection process by other microscopy techniques. So here I show you a section of a nodule at 3 weeks post-inoculation with IRBD74. And in purple, you can see the colonization of the nodule cells, stained with color blue, and they are very well colonized by IRBD74. And when we observe in more detail this part with the scanning electron microscopy, you can see very clearly here between these two epidermal cells the infection, the intercellular infection of IRBD74. here is the intercellular space. And then we analyzed another region of this nodule in this region with cortical cells. And once again, you can see in the intercellular space the deposition of IRBD74 all over. And in the nitrogen fixing zone, we also see the release of IRBD into the nodule cells from these infection beds that are intercellular infection structures. So in general, with all these different techniques, we show that IRBD was able to nodulate lotus and that this infection was via intercellular colonization. And then we were wondering if this intercellular colonization requires a different genetic program. And in the last decades, different research groups have identified different genes, plant genes, legume genes, that are very important for this process, for this integration process. And mutants affected in all these genes are also affected in rhizobial colonization. So we decided to analyze the symbiotic performance of IRBD74 with each one of these mutants. And I will not show you all of them now, but I just want to highlight two of them. Is this receptor, kinase, and this transcription factor. And the reason of why I highlight these two ones is because we already know that these mutants with mesorhizobium loci in the intracellular colonization, reader K1 only develops white nodules, which indicates that it's a deficient rhizobial infection process. And in the case of R1 develops very few nodules and with a delayed nodulation program compared to the white type. However, when we tested these mutants with IRBD74, the symbiotic phenotype was not so strong, and they perform very well, actually. So you can see it's almost the same size of this nodule in R1 than in the wild type with IRBD74. And when we analyzed the rhizobial colonization by qPCR in these roots, we confirmed that there was more colonization by IRBD74 in these mutants than with R7A, with mesorhizobial loci. And it's the opposite in the wild type. So here I just forgot to mention that IRBD, yes, makes nodules, but the nodulation program is a bit delayed. So that's why we have more colonization in wild type conditions with M-type than with IRBD74. And then these results was confirming that there is a different genetic requirements for this intercellular infection process. So we decided to explore this in more detail, and for this we made RNA-seq. So we inoculated Lotus japonicum roots with IRBD74, and we collected the roots at 3, 5, and 10 days post-inoculation. And we chose these time points because, as I told you, the rhizobial and organogenesis program is a bit delayed. So we wanted to compare with 1 and 3 days post-inoculation with M-type. That is a bit equivalent in terms of rhizobial infection than with IRBD. And as you can see here, there were many differentially expressed genes with both symbionts, and there were many genes that were shared between these two. However, there were some genes that were only differentially expressed with mesorhizobial loci, and others that were only differentially expressed with IRBD74. And another important finding was that all the relevant symbiotic genes were induced by IRBD74, as it occurs with M-type. But one of the most interesting findings that we have with these RNA-seq experiments was regarding cytokinin-related genes. So we found a very different expression profile of different cytokinin-related genes. And you can see in this heat map how different is the response with IRBG and endotype. So with this interesting finding, we decided to explore this in more detail. And we tested several mutants affected in cytokine or cytokine-related mutants. So we have two genes involved in cytokine biosynthesis, and this one is a cytokinin receptor. So it's important to mention that these mutants, they have been already tested with mesorhizomal loci. And in the case of LHK1, there is a delay in nodulation, but they can form nodules. However, with IRBG74, the mutant was unable to form any nodules, even not bumps. And with the other two mutants, there was a strong delay and reduction in nodule formation. And here you have some representative images of the nodules that were formed at 3 and 5 weeks post-inoculation in these mutant backgrounds and compared to the wild one. And this, of course, has an impact in the plant growth when we harvested the experiment at 6 weeks post-inoculation. So the growth was really compromising these mutants compared to the wild one when inoculated with IRBG74. Well, in this part of my talk, I show you that we developed a novel working model to study intercellular infection with Lotus and IRBG74. We also show that IRBG74 induced a distinctive cellular and transcriptional program, where some genes were more important for the intracellular colonization with endotype, but other genes, like the cytokine-related genes, were more important for the intercellular infection by IRBG74. And we have a core of genes that are equally important for intracellular and intercellular colonization. And this finding was very important for us and for the field, I think. But then we were wondering if we could identify novel players. And in order to identify novel players in the intercellular colonization, we decided to employ comprehensive approaches. And I will show you some examples of the different approaches we have employed and some of the results I have obtained. So the first approach was a mutant screening. And in Lotus japonicus, we have Lotus retrotransposon mutant population. And this was derived from an accession of Lotus japonicus with epigenetic activation of the LOR1 element during the germline. So the result is that each seed coming from this accession will have different retrotransposon insertions in the genome. So as you can imagine, there will be different genes affected by these retrotransposon insertions. Usually we have between 4 and 6 insertions in each mutant. And because we know the sequence of this retrotransposon, we can track the insertion and identify the gene that was affected. And there is also another interesting aspect of this one. This LOR1 mutant population we have. These are natural mutants. So these are not GMOs, so it's very easy to work with them. And well, so the experimental strategy we developed was to germinate around 2,000 LOR1 seeds. And then we put the seeds, well, I put the seeds in these pots. Then I inoculated the seedlings with IRBG74, and I harvested the experiment between 6 and 8 weeks after inoculation. And when I harvested the experiment, I collected plants with a not minus or fixed minus phenotype. And the fixed minus phenotype means that there were white nodules. And white nodules means deficient rhizobial colonization. So the plants that were selected were sent later to the greenhouse for seed production. And once we received the seeds, I made an nodulation test on plate for each of the mutant lines. So for each mutant line, I was testing about 10 seeds of the offspring And it was to confirm the symbiotic phenotype. So here you can see in this example how many of them were yellow, which is a symptom of a deficient symbiotic process. Well, so I repeated this process many, many times until I tested 200,000 plants. And from this amount of plants that I tested, I selected around 1,200 mutants, completely different mutants. And from these 1.2 thousand mutants, I made nodulation tests on plate for almost 600 different mutant lines on plate. And well, I could confirm the symbiotic phenotype in more than 100 mutants. And we started to analyze these mutants. So for 50 of these mutants, I follow all this protocol to identify the LOR1 insertions in the genome. And well, in 34 mutants, I could identify new LOR1 insertions. These LOR1 insertions were distributed in the different chromosomes in this pool of mutants, some in the intergenic, intron, promoter, or in the exon. In total, they were almost 200 LOR1 insertions in these 34 mutants. And here is just a representative example of each of the mutant lines with the different insertion and the positions observed. So in total, almost 90 LOTU genes had at least one LOR1 insertion in this collection of mutants. And well, just here I show you that when I analyzed the genes that were affected in this pool of mutants, some of the genes had a very interesting expression profile in the Lotus expression atlas. So they become, or they are very good candidates of the symbiotic phenotype that we have observed. And here are some representative examples of the different mutants I have and with the symbiotic phenotypes. So I have some mutants that are nodulating very well with Mesorhizobium loti, but not so good with IRBD. Others are not good with any of these rhizobial partners. And I also found some not minus mutants. But one of the most interesting mutants I have, I call it the intercellular deficient mutant. And it's very interesting, this mutant, because with R7A, is able to form practically normal nodules to similar numbers than the wild type. However, with IRBD74, it's completely not minus. There are no nodules at all and not bell bombs. So I am characterizing now this mutant, and then I was wondering if this symbiotic phenotype was associated only to the intercellular infection with IRBD74 or to other kind of symbiotic association. And here, just briefly, I tell you about the arbuscular mycorrhizal symbiosis. So in this case, the spores germinate in the epidermis of the roots. Then hypopodium is formed. Then some hyphas that starts to colonize the epidermal and root cells and cortical cells. And eventually, arbuscule is formed in the cortex, and the symbiotic association is established. Well, so all this process could be was nicely visualized in the wild type. So here I show you confocal microscopy image I took from lotus root inoculated with Rhizophagus irregularis and stained with WGA to visualize the different AM structures. So you can visualize all the different structures. However, at this time point in the intercellular deficient mutant, I see a very big and spread hypha formation, but very few arbuscules. And here are the graphs that show these observations. So I am still characterizing this mutant. And what is very interesting is that none of the non-symbiotic genes that we know in the field is affected in this mutant. So it seems that it's a novel player. And well, now I will show you a different approach I have employed, and it's through the transcriptome comparison of intercellular and intercellular colonization. So from the data I showed you before, from this RNA-seq analysis, so I found two different and interesting aspects. One is that there were some genes that were only upregulated by IRBG74. And also that there were many cell wall related genes highly induced by mesorhizoidal and IRBG74. So now I am making different constructs to analyze the promoter activity of these genes. And I am also obtaining homozygous mutants in all these cell wall related genes. But this time I will send you some results I have regarding this part. So I obtained homozygous mutants for each of these genes in Lotus. And in 3 of them, there is a good symbiotic phenotype. An expansion, this cytokine-related gene that I showed you before, and aldolase also. And here I will present you some results I have with the aldolase. So this aldolase is highly induced by IRBG74 at 5 days post-inoculation. And then I decided to obtain some homozygous mutant, and I got 2 different mutants, so 2 alleles in different positions. And the aldolase is an enzyme that is part of the chorismate pathway for the biosynthesis of these aromatic amino acids. And when I observed the mutant, immediately I found a very interesting phenotype. Because the root hairs emerge and develop like normal, but at some point they start to get swollen and they start to have these balloon-like structures. And finally, the root hairs collapse. And I observed this in the 2 different mutant alleles. So I decided to analyze more in detail this mutant. And when I checked the 2 different alleles with IRBG74, I observed a drastic and significant reduction in nodule formation, as you can see here. And here there are some representative examples of the nodules at 3 weeks post-inoculation in the 2 different mutant alleles. And because of these results, I decided to do other experiments to characterize this gene. So here I show you some Lotus japonicus hairy roots expressing the promoter of the aldolase fused to the triple YFP with a nuclear localization signal to analyze the expression profile of this gene. So you can see a high expression, a high promoter activity in the meristematic region and in different root tissues, including the root hairs. In response to MLOTI and to IRBG74, we could see the promoter activity in the epidermal cells and also in the cortical cells, as you can see. And also here you can see very nicely root hair infection thread with mesorhizoidal. And here during these infection pockets with IRBG74 in the epidermis and cortical cells. Then I wanted to check the subcellular localization. So I made this construct with the promoter of the gene, the CDS, and then fused to the YFP. And we observed this chimera in structures that resemble plastids. And you can see all this localization in these structures. And here also what I want to highlight is that this was done in the aldolase mutant background. So we were able to complement the root hair phenotype. And well, here also I am characterizing this mutant, and there are other experiments, but these are just some examples. And well, the third approach I have been employing is a genome-wide association study of intercellular infections. So in Lotus japonicus, we also have more than 100 different ecotypes. and we have genome information for all of them. And they belong to different regions of Japan. So we were wondering if these different ecotypes have different intercellular infection capacities. So I inoculated these more than 100 ecotypes with emlotype or with IRBG74, and then I counted the numbers of nodules at 6 inoculation. And indeed, I found very strong differences in the nodulation capacity with IRBG74 in some accessions. So this is a representative example of one of the accessions that have a very poor capacity to be nodulated by IRBG74. So here you can see the poor growth of the plant. But with GIFU, that is the accession that we use in the lab, IRBG is able to grow, well, IRBG is able to promote a good growth of the plant, as you can see. So actually, most of the accession that I tested have a lower capacity to be colonized by IRBG74 compared to GIFU. And well, then with the help of our bioinformatics unit, we performed the GWAS. So we use all this data in the Lotus GWAS portal developed by Steve Anderson. And they perform these Manhattan plots to identify some potential peaks and the SNPs associated. They make more bioinformatic analysis. And after all this analysis, we identify more than 80 different SNPs or potentially, with more than 80 different genes potentially linked to these SNPs. And 50 of them are genes of non-function. So we performed more analysis to try to select which ones we will choose. And we chose these different genes. And I obtained homozygous mutants for each of them. And then I tested the nodulation fit phenotype for each of them. And here I show you also the expression profile of all these genes, and we chose them because they were either induced during the root nodule symbiosis, during the arbuscular mycorrhizal symbiosis, or even with some pathogens. So some of them, they did the data nodulation phenotype. And I am now focusing in some of them to try to characterize these different genes. And here is just a representative example of 2 of them, because both of them are peroxisome-related genes. You can see that the homozygous mutants have a lower nodulation rate compared to people, and also reduced growth when I harvest experiment. So, this is the first part of my talk. And as a summary, I just present you here that I employ different approaches to try to identify novel players in the intercellular colonization between Lotus and IRBG74. I am trying to characterize different genes, and I consider that to accomplish this, they can be divided in short, medium, and long-term perspective. And now I will move to the next part of my talk. And this is the research I made in the group led by Professor Iva Kondoroshi in the Hungarian Academy of Sciences in Szeged, in Hungary, and with the collaboration of other professors and PI. And well, in this part of the talk is more focused about the nodule function and in late stages of the symbiotic process. So as I already told you, there are 2 types of nodules, the terminate nodules and indeterminate nodules. In the case of Lotus japonicus has the terminate nodules, like being also. So, and there is only one nitrogen fixing zone, and they are round shaped, as you can see here in this row. By contrast, the other model legume, Medicago truncatula, has indeterminate nodules with different zones, like the apical region, the infection zone, interzone nitrogen fixing zone, and also a sensing zone. And there are also other differences between these legumes. Because as I told you, once the bacteria are released from the infection threads in the nodule cells, they become bacteroids. And bacteroids can be terminally differentiated bacteroids or non-terminally differentiated bacteroids. So in the case of lotus, Phaseolus vulgaris, and soybean, and other examples, the bacteroids have the same size than the free living culture. They maintain their DNA content, and they also keep their reproductive capacity. So when we isolate the bacteria from the nodules, they can still divide. In the case of Medicava truncatula and some closely related species, the bacteroids, they have a drastic change in their morphology, like these examples. They also increase their DNA content, but they lose their division capacity. So if we isolate these bacteria, only very few of them will be able to form colonies. And this can be observed in Medicavo, in Pisum, and some closely related species. So then we were wondering if this process was only present in Medicavo truncatula. Because in Medicavo truncatula, the laboratory of Iva Kondoroshi showed that the terminal differentiation is because of these nodule-specific cis-enriched peptides. And these NCRs are a big family, about 700 members in Medicavo truncatula. And it's interesting because they have antimicrobial activity, and they are only expressing the nodules. So we were wondering if this process is only in Medicavo truncatula or in other legumes. Because we already knew that all these species that belong to the visual clade, they have NCRs, and the bacteria were branched and elongated. Even one from this other clade, the astragallian clade. However, we didn't know anything about some basal legumes from this IRLC. So we decided to analyze if bacterial differentiation was something in all these different species from this clade, and also what is happening with the NCRs in these different species. So to start to answer these questions, we decided to inoculate these legumes with their rhizobial partners and check the morphology of the different bacteria. And when I collected the bacteria from these different nodules, from these different species, different subclades, you can see that it was very interesting that they have super different morphotypes of bacteria. Some of them were elongated and a bit branched. Others were slightly elongated, spherical, and rod-shaped. And then the next question was if these different morphotypes is part of the differentiation process of the bacteria. So as I told you before, the terminal differentiation of the bacteria is characterized by 3 different aspects. And one of them is that the bacteria have an increase in their size. So I measured the size and area of the different bacteria, and I compared this size with the size of the free living cultures. And in all the cases, we observed significant differences, an increase in the size of these bacteria compared to the free living cultures. So another feature of terminal differentiation of bacteria is that they lose their reproductive capacity. So to test this parameter, I isolated the bacteria from these different nodules from different species, and then I analyzed it by colony forming unit assays. And as you can see here, all these legumes, all these bacteria, lose their reproductive capacity. And this contrasts so much with soybean, where they don't have terminal differentiation of the bacteria. Also, I tested another parameter, that is the DNA content, and all of them have increase in the DNA content. So we concluded that all these bacteria, all these species, impose terminal differentiation in the rhizobial partners. And well, the next question was if all these different species have NCRs or not, because we knew for some of them. So to answer this question, we collected again nodules and we made RNA-seq from the nodules. And then we tried to identify NCR expression in these different nodules from these different species. And the answer is yes, we found NCRs in all the species that we analyzed. However, it was very different, the number of NCRs that were expressed. So in Glyceris ouralensis, which is here, we only found 7 NCRs. But in the case of Meditado, Pisum, and Galega, we found hundreds of NCRs. And here also the interesting finding is that those species with elongated and branched bacteroids have more NCRs. And we found a correlation between the number of NCRs and the increase in the cell size of the bacteria. So there was a positive correlation there. And the NCRs are small peptides, but they have different physicochemical characteristics. And one of them is the isoelectric point. So they can be divided into anionic, neutral, or cationic NCRs. What we found is that those species with elongated and branched bacteroids have a higher proportion of cationic NCRs. When we check the expression levels, we also found that these species with elongated and branched bacteroids have a higher expression of the cationic NCRs in this heat map. So these different results were telling us that the isoelectric point of the NCR was something important and related to the morphotype of the bacteria that they were causing. So then we decided to test NCRs with different isoelectric points in free living cultures of Ensifermeliloti. So we have different NCRs, synthetic NCRs. And here you can see that the anionic NCRs, they produce in Ensifermeliloti this biofilm-like structure with extracellular material. In the case of the neutral NCRs, they also form some kind of biofilms and extracellular material, but it was different from the anionic ones. And with the cationic NCRs, we observed this vesicle formation and even some morphological changes. And when we observed this, it was something very interesting because when we checked the bacteroids inside of the nodules without any treatment, so only the bacteroids, we saw that the bacteroids from Galega orientalis and Medicago truncatura had this kind of vesicles on the surface. And these two legumes have high expression of cationic NCRs. In contrast, Glyceris ouralensis, the one with only a few NCRs, doesn't have any cationic NCR. And the surface of the bacteria was without these vesicles. By curiosity, I treated Ensifermeliloti cultures with a combination of different cationic NCRs. And I observed some elongated and branched Ensifermeliloti cells with these vesicles that resemble so much the bacteria. So this was the research I made in Hungary with Ivan Kondoroshi, and we could publish the results some years ago. And well, finally, in my third section, what I have shown you so far is that using these two different model legumes Medicago truncatula, and Lotus japonicus. We had very interesting findings and novel discoveries. However, I think that now with this knowledge, we can start to explore plant microbe interaction in legume crops, like peanut and bean. And I will present you why I think that these two crops are very important to do research in the legume rhizobia symbiosis. And in the case of peanut, well, peanut is a very important crop, also here in Morelos. There is something interesting with peanut, that is that peanut has intercellular infection. And this is the process I have been studying in the last 3.5 years. Another interesting aspect of peanut is that peanut has these spherical bacteries. And this spherical bacteries is an indication of terminal differentiation, like the ones I observed in Amanus spinosa. And then here, it is possible that there is terminal differentiation and SCR-like peptides. And this opens the possibility to explore antimicrobial peptides in arachis. And another point that is very important is that there are already protocols for heavy root transformation, so we can start to explore it. And also the genomes is available of this legume. Well, regarding Phaseolus vulgaris, so there are some biological questions that we can answer with Phaseolus, because it was shown here in Mexico by I think here's visible the name of Esperanza, that Phaseolus vulgaris is able to establish symbiotic associations with a broad range of bacteria, of rhizobial partners from different genera. And this is something that we are starting also to explore in my current group, to try to identify genetic elements in legumes that allows a broader host range. Because this has very important implications in agriculture when farmers try to implement some legume crops in soils where there are no native rhizobial species. Another interesting finding was when I was writing my article about IRBG74 and Lotus. Ewan James, our collaborator, he gave us some references about IRBG74. And when we were writing the paper, we were wondering if we say that it's an agrobacterium or a rhizobium. And we refer to the findings made here in the CCG by Jaime Mora. And it's very interesting that Agrobacterium fusense, a closely related species to IRBG74, was found in bean nodules. So I am about to test if IRBG74 is also able to colonize Phaseolus vulgaris. And well, another important aspect is that in Phaseolus vulgaris, there is genome available. There are transcriptome resources developed by genome. There is heavy root transformation that was found by Federico Sanchez. So we have different tools that we can study very well the Phaseolus vulgaris rhizobia symbiosis. And finally, I would like to emphasize two different aspects. One is that Phaseolus vulgaris and rhizobia symbiosis is the working model that is used by different groups here in the CCG. And actually, it is the working model I used during my master and PhD, so I have a very good knowledge about this process. But I think that it is very important that in the research center, you have the possibility to exchange ideas and with different angles from the bacterial and plant perspective. And then you can see only here all these different publications about the Phaseolus vulgaris rhizobia symbiosis made by the CCG and by the campus here in Cuernavaca. So I think it's very important to maintain this leadership in the Phaseolus vulgaris rhizobia symbiosis because only few groups are studying this process. And especially Physiolus vulgaris is highly relevant for Mexico. Because Physiolus vulgaris is the center of a region here in Mexico. So we have a rich pool of natural variation, and we can start to answer several relevant biological questions, exploiting this natural variation that we have here. And now with the new technologies that we have developed. So so finally, I show you here in my presentation that we can use the knowledge in Medicago truncatula and in Lobfuture conifers to start to expand this knowledge to other crops like arachis and Physiolus vulgaris. And using this collaborative network that I have developed in the different groups where I have made my research. And the main focus of my proposal is about intercellular colonization or intracellular colonization. However, I think we can start also to answer other questions. And I have a special interest in the interplay of cell wall related genes during rhizobial infection. And well, as you know, this program, Functional Genomics of Eukaryotics, has researchers that are focusing on microbial. So I think that it will be very interesting to test some of the genes I identified in my intercellular infection process, because not most of them, but many of them, I found that they were also induced by RNA-seq by some pathogens or different pathogens. So this is a very interesting aspect that we can start to explore. And also, when I was checking more about peanut, I found that Damien, he already found some microRNAs in a collaboration with a group in Argentina. So I think we can start also to explore the regulation of this intercellular infection process, no, by other mechanisms. And finally, I think that knowing this intercellular infection can start to give us some knowledge to explore other aspects, like the microbiome composition. And microbiome and endophytic composition is something we really think we really should start exploring in Physiolus vulgaris, because there are currently a lot of grants about these aspects, because it has been shown that this can boost the plant growth. And well, finally, just to emphasize how relevant is still this field, the legume rhizobial symbiosis field, I wanted to share with you a video that was made by the European Research Council about legumes. So I hope it's going to work. I think. Okay, and with this is the end of my talk, and I am happy to hear any questions from you guys. Thank you so much.
Thank you very much. Differential response. Intercellular.
Perdón, ya no se oye lo que están diciendo.
Yeah, I think that's a relevant point. So it's possible that in the list of genes, there are some of them, but also it is possible that the time points we check probably were not the best to start to see these responses. I mean, these responses of the expression of these genes. But yeah, I mean, you're right. However, with the other approaches we employed, I could find some genes that were identified as important for intercellular infection, and they were induced during pathogen interaction.
So perhaps the other side of the coin would be to study process of the story, because it may be masking the response from the process. Only one way to understand the progressing without currently listening strong, but visually evaluate.
Well, actually, I mean that's completely true, and actually that's why one of the projects of one of the postdocs is he's already producing a mutant population of IRBB74, and we are about to test this mutant population of IRBB74 with Lotus japonicus to try to identify the bacterial components that allow this intercellular colonization. Thank you.
Perdón, ¿podrían repetir las preguntas antes de las respuestas? Porque no se entiende nada lo que están preguntando.
Ok, sí.
Bueno, entonces cuando habla de Jesús, the strength of these is what they belong because you mentioned that I don't know if you mentioned what the below this strength.
Well, the way they mentioned in the paper of Jaime Mora, they oh yeah, sorry. Yeah. So the question is to know how do we know if it belongs to a Agrobacterium or Rhizobiasis?
Right? You were saying
the strain IRBB74. So that's the question.
Yeah.
So we refer, I mean, what we found were papers, already published papers, where they refer that this strain is actually an Agrobacterium strain that contains some symbiotic sequences. And it's closely related to Agrobacterium crucense. So it's located as an Agrobacterium species belonging to the Rhizobiasis. That's the classification they report.
Okay, thank you. And regarding this, could you your opinion about could you for example, your opinion
which kind of infection is more efficient, intercellular or intracellular? In your opinion, regarding specifically in terms of the regulation and acquisition, etc.
Okay, yeah, so the question is about which type of infection process is more efficient,
In your opinion,
in my opinion, the intercellular infection or the intracellular infection. Exactly. So the evidence that we have collected and the discussion is that intracellular infection seems to be more evolved and more efficient. But because we can find this intercellular infection only in some basal layers. And that's why we consider that the intracellular colonization is more evolved and more efficient. And there are several molecular players that it seems that they were evolved to allow this intracellular colonization.
One is called this kind of infection because this infection in protein is less efficient than the one.
Exactly, yes. I mean, it's less efficient, but it's more simple. And then if we want to reach rhizobial colonization in non-legume crops, we have to start with the most simple methods. And because this intracellular colonization requires this infection transformation, which is a very complex problem, then it will be just easier to try to identify, in our opinion, the players that are important for this intercellular colonization without triggering this different response. Because then we can reach rhizobial colonization in some non-legume crops.
Okay. Thank you, Jesus. Very nice talk. I have two questions. Actually, one of them related, as Sergio mentioned. You never showed the efficiency of assimilation of nitrogen assimilation in the IRAG 74. And as far as I understood, so it should be less than the MLOT, right?
Yes.
Okay, that's okay. But the other question that I have is, you mentioned that actually IRAG 74 interacts with Arabidopsis, right? Do they have any phenotype, any improvement of the on the development?
Yes, exactly. Yes. So it was not naturally found in Arabidopsis. It was naturally found in rice.
Mm-hmm.
And then because they found it in rice and also with Sesbania, then they tested in Arabidopsis. And they found an increase in the root architecture development system. And they speculate that they induce toxin production.
Do you have any plans to work with Arabidopsis and to try to answer, because as a model,
Yeah.
try to answer any question that
I unfortunately haven't worked really a lot with Arabidopsis, but I think it will be very important to start to do some stuff with IRBG 74 in Arabidopsis. And actually, some of the colleagues we have in the ENSA project, he's already testing IRBG 74 in Arabidopsis and using some markers of toxins.
Okay.
And also, some researchers are testing IRBG 74 in barley.
Mm-hmm.
So, because this has this very high capacity to infect and to colonize roots of legumes and also non-legumes. So, I think, through collaboration, it would be very nice and interesting to test this in Arabidopsis.
And what about, so do you know if they have any biostimulant effect of this IRAG 74 in relation to defense response? Is once they interact with Arabidopsis, I mean, on other plants, and then once they are interacting the roots, they can confer some defense response in the upper part of the plant. Do you know that?
I mean, that's also a very, very interesting this trend, because I mean, I tested once with the medical, and the plant looked very sick. So it was more like a pathogen plant, no? So, I mean, we haven't tested with different plants, no, this IRB. We have tested some, not with another. And we haven't checked, like, in detail, this different response, and why some plants are happy and are welcoming IRB 74, but others, they don't want, no? So it seems that IRB 74, it has this symbiotic capacity, but also it can act somehow like a pathogen.
Okay, yeah, thank you.
I was going to ask, but you have already mentioned the possibility or the hypothesis that this kind of intercellular colonization of this Rhizobia strain would be important, can be important for colonizing other plants, non-legume plants.
Yes.
So now you spoke about this in Arabidopsis to Mario. Do you know anything about other like Graminia or
I mean,
regarding this strain?
yeah, I mean, we have tested with some few plants, like Americano, we tested with barley, and other tested with barley. We know the evidence in rice and Arabidopsis. But I think, I mean, this strain is super interesting, and I think it works best trying on other like crops and other plant species. And I think eventually it will be also very good to have this mutant population of IRDB 74. And to start to test this mutant population with also different crops and non, yeah, especially crops. Actually, one of the ideas I have is to test this IRB 74 with different Phaseolus vulgaris taxonomy. And this is something that also about my perspectives, that we can start to explore and to exploit really all this natural variation with Phaseolus. Something similar to what I have done in Lotus with this 100 or more than 100 ecotypes. And I observe differences. Even if, I know, Japan is not that big like Mexico, no, in variation, and still you can have a high diversification in the capacities of some ecotypes. So that's something very relevant that we can start exploring.
Yeah. Another question that I was just thinking, there are some rhizobial strains that are promiscuous. One of them is the NGR 234.
Yeah.
And it has been shown that the capacity, in some cases, the capacity to nodulate different legumes of this strain is related to effectors that the bacteria synthesize and excrete.
Yes.
And then if with mutants in this kind of genes, this capacity is somehow blocked.
Yes.
Do you know if the IRBG has also effectors that would be helping the association with plants or no?
I really don't know. So we haven't explored this aspect. I mean, recently what we are trying to explore in Lotus Bhutan is this promiscuity. Because usually, yes, what we know so far, most of the knowledge about the promiscuity in this Rhizobia legume interaction is coming from the bacterial side, practically most of them. But we don't know very well the determinants from the plant side. We know now recently that in the case of Medicago concatura, part of the compatibility comes from these NCRs. So there was a publication from Eva also and another person that they found that these NCRs also define the compatibility between symbionts. But in legumes without NCRs like Lotus or like Phaseolus vulgaris, I think it should be very interesting to start exploring what are the genetic components.
Pues, vamos a intentar que hay una pregunta de Luis Cárdenas. Vamos a ver si la ¿Puedes subir el volumen? Luis, si quieres, empieza a hablar y vamos a ver si Jesús lo escucha.
Gracias. ¿No había una pregunta antes de Esperanza?
Pues Esperanza ya se desconectó.
Okay. So, it's Jesus. I just wanted to ask you about I was really surprised about the transcriptomic analysis that you were doing, comparing the colonizing bacteria by the intercellular colonization and the other one that is the traditional ones. But when you look at the expression of genes involved during the intercellular colonization, I don't see that much of genes involved in cell wall remodeling. You know, it seems that are more important in the intracellular colonization than in the intercellular colonization. Do you have any explanation for that? I mean, I was expecting to see more enzymes involved in cell wall remodeling because they have to go through this intercellular space, like remodeling all this space, but it doesn't seem to be the case.
Well, hi, Luis. Thank you for your question. Okay. Yeah, thank you for your question, Luis. Yeah, actually, I didn't find like much more cell wall related genes with IRBG than with mesorhizoidal. However, what I have so far is some other kind of evidence. So I have two different mutant alleles, well, two different mutants in two different species. And these two different mutants have a stronger phenotype, symbiotic phenotype, with IRBG74 than with mesorhizoidal. So even if I didn't find something by the RNA-seq approach, the homozygous mutant seems to be more affected in the intercellular colonization with IRBG74. Also, for instance, this non-peptide liase that it was published several years ago, this also has a stronger phenotype with IRBG74 than with mesorhizoidal.
Thank you, Jesus. Just very briefly, my second question is the number of NCR peptides that you were talking in the second part of your talk. You were saying that, well, this is very, you can see that there is like a gradient, like some rhizobia species that express very low number of these NCR peptides, but there are some species that have this high number of these NCR peptides. Do you know if there is any correlation between the number of these NCR peptides, because they get all these morphological changes, and the nitrogen fixation efficiency? Do you know if we have like any correlation between this, you know, these both phenotypes?
Well, I mean, regarding this, I think it's very difficult to compare between different legumes because the nitrogen requirements can be very different. So the comparison is a bit tricky. However, there are some reports I remember where they compare the acetylene reduction or the capacity to convert to reduce, I mean, acetylene in a combination where they use the same bacterial species that was able to colonize two different legumes. If I remember correctly, one is Pisum sativum and the other was a legum with a non-terminal differentiation. What they described and they reported is that it seems that in the bacteroids, the terminal differentiation was higher acetylene reduction. But yeah, several researchers are not very happy with this kind of comparisons.
Okay. Thank you, Jesus. And just a very, very short comment at the end. I just want to tell you, I think that is very interesting from your work, this cytokinin signaling that you see in this intercellular colonization, because I think that that could be very important from the evolution, you know, of the symbiosis. It's very interesting to me that is more dependent on the cytokine signaling than on the regular one that has been described in the other kind of colonization. So in this regard, do you think that you didn't talk about that, but the number of nodules that can be induced by this new way of colonization is similar to the traditional one? And if you could consider that the autoregulation of the nodulation will be like different or differentially adjusted?
Well, I think that's a very important point, Luis. So regarding the auto-regulation of nodulation, I did notice that some of the clade peptide or clade genes, they were slightly more induced or differentially induced by IRBG74 and by Mr. Sobuloka. And yes, I didn't show you the total number of nodules and the comparison, but it's very similar when I harvested the experiment. So at 4 to 6 weeks post-inoculation, the number of nodules were similar. But I did notice at early time points that the transcriptional response of these clade peptides was a bit different with the different rhizobial partners. And regarding cytokine, yes, I mean, now it's a very interesting point because, you know, there are some recent publications about the interplay of some phytohormones necessary for lateral root emergence and also for nodule organogenesis. This is some research that is conducting JAX already. But yeah, I think this binding with cytokine is something we could try to explore a little more in detail. But working with phytohormones is very tricky also. It's not that easy.
Thank you, Jesús.
Bueno, tenemos aquí otra pregunta en el chat que te voy a leer. Could you recommend bioinformatics approaches to identify NCR genes?
Well, the way it was done, it was by a bioinformatician we had in Eva's group. And the way we did it, or he did it, is that for most of the language, we were trying to identify the NCRs. So there was no genome available or sequence genome available. So what he did was he used this, I think it's called Trinity software to try to assemble the transcripts. And then we were searching for certain numbers of cysteine residues, and the distance between them. So there are some reviews, some publications from Eva and Peter groups, where they define very well what is the distance between these cysteines. And also the other way to find them is that they have a signal peptide. So these characteristics and the presence of an intron, in the case of Medicago and closely related species, was the criteria used to identify these NCR peptides. But I mean, if you are really interested, I can provide you the information in more detail.
Okay. Related to this, I have a question. So I was was striking to see the numbers of these NCR genes in different genomes, from a few to a few hundreds. And where I was wondering, where are they encoded, or do they form clusters in the genomes? I mean, it's like when you have 700 in Medicago, is it like you have clusters of many genes, or is it like?
Yeah, there are tandem of genes also. But there is actually in all the different chromosomes, as I remember. Maybe there is something interesting about the analysis we made with these NCRs in the different levels. One very interesting finding is that if we analyze these 7 NCRs of Lysia uralensis, practically 6 of them are identical in other languages, like the closely related legumes of the most basal ones. So then it was an indication that they were maintained during evolution in these other legumes. But these 6 or 7 NCRs, we couldn't find them in Medicago truncatula or Pisum sativum or Medicago sativa. So they were completely different. And even when we compare Medicago sativa and Medicago truncatula, all the 600 or 700 NCRs, and we make classes in them, they were not putative orthodox. So the sequence was super different. I mean, all of them were NCRs, but the sequence was very different. So it seems that the evolution in this more evolved language probably was very fast for these NCRs.
See, thank you. Here's another question from Luis Cardenas. He is asking, is intracellular colonization only limited to determinate nodules?
No, it is not, because we have, like, I mean, Medicago species, Pisum sativum, and many others. So all the mixture plates have indeterminate nodules, and they have intracellular colonization.
So, is there other question? More question? In the chat seems to be empty. And okay, there's one.
How general do you consider your model regarding the model that you saw us specifically the role specifically some pathways, some players for some cytokinins? What do you think is general in regarding to another species of bacteria made this kind of intercellular infection?
That's a very good point. And this is something now, I mean, when we released this publication, it was like a very, like the first publication with a lot of gene players in this intercellular infection, right? But after a few months, there was a publication from a French group where they analyzed Skinomene, and Skinomene has this intercellular infection process. And Skinomene has only intercellular infection. No? Lotus has intra and intra. And they found some overlaps, like the ones we found, the same findings. But also what they found is that there were, for instance, in Skinomene, there is no a protein called RPG. And we found that in our system, RPG is still expressed and still slightly important. So, well, the conclusion of this question is that it seems that the intercellular infection process or program, they have many overlaps with other legumes that are intercellularly infected. But also we can observe some differences. So now we are starting to write a review with the guy who is now actually in the laboratory now, the guy who published this in Skinomene. Because yeah, we have found that there are some similarities or many similarities in the genetic program, but also there are really important differences.
Okay, but we have one more question. Yes, but well,
Nice presentation.
Thank you so much.
You mentioned your interest in microbiome and endocytic composition in this. What do you think about the one work released in December by Mora Groups, reported a consortium of bacterial insects of isolated species? What do you think about it?
Sorry, the last part it was.
This group report a consortium of bacteria in Rhizodium N6.
Okay. Okay. Well, I actually don't have yet so much knowledge about microbiome association. So currently we are starting to do that. I mean, in my case, currently I am starting to test my different mutants with the synthetic community we have in Latin America. But I think this is really something we have to start to develop a synthetic community in being and start to analyze the composition of this synthetic community and also in different accessions. That's my opinion.
Okay, so I think now we have answered all the questions. Thank you very much, Jesus, for the nice presentation. Very interesting. Thank you to all the people who assisted today here in the first time after, well, since 18 months or more, we are like in the same place. And thank you to all of you who followed by Zoom and by YouTube. And we'll see you next time. Thank you very much.